Ever really thought about what goes into making the medicine in your cabinet? I mean, it's way more than just mixing a few ingredients together. Today, we are diving deep into a process that's absolutely crucial but almost totally invisible. It's called size separation, and it's the hidden science that makes sure your medication is actually safe and effective.
So, to kick things off, let me just ask you this. Picture you're working with a medicinal plant, grinding it down into a powder. Why would something as simple as sifting that powder be, well, incredibly dangerous?
Believe it or not, the answer is because you could literally be throwing the most important part of the medicine right into the garbage. Seriously, the stakes here are sky high. If you get this process wrong, a drug could become completely useless, or even worse.
And that really gets to the heart of our explainer today. Why on earth does the size of a tiny little particle matter so much, especially when you're dealing with powders?
Okay, first things first, let's get a clear definition. Size separation is basically the process of sorting a jumbled-up mix of particles into neat groups of a specific size. You've probably heard it called sieving or sifting or screening. It's all about bringing some serious order to the chaos of a mixed powder.
Right. Let's go back to that dangerous plant example. Here's the deal. A plant isn't the same all the way through, right? The soft, leafy bits, they grind up super easily into a really fine powder. But the harder, tougher parts, like the veins, they stay in bigger pieces, what are called tailings. Now, in a drug like digitalis, the active ingredient, the real medicine, is packed into those tough veins. So if you just sift the powder and chuck out the big bits, you are literally throwing away the medicine.
To prevent that kind of disaster, the pharmaceutical industry doesn't leave anything to chance. Oh no. They have very, very specific rules. We're about to look at the official powder grades that bring some much-needed precision to this whole process.
So, check out this table from the Indian Pharmacopoeia. It shows 5 official grades of powder, everything from coarse down to very fine. But what do all those numbers, 10, 44, 60, actually mean? It's not just one simple measurement, it's actually a clever 2-part test. Here's how it actually works. Let's take that coarse powder, which is also known as a 1044 powder. Okay, so first, every single particle has to be small enough to pass through a number 10 sieve. Simple enough. But, and here's the key, to make sure that powder isn't too fine, there's a second rule. No more than 40% of it is allowed to pass through a much finer number 44 sieve. See? This 2-step process basically guarantees the powder is locked into a very specific size range. Pretty smart.
Okay, so we keep talking about sieves, but what exactly are they? I mean, we're not talking about your kitchen strainer here. Let's look at the tools of the trade and really break down the anatomy of a proper pharmaceutical sieve.
The most basic thing to know is the sieve number, and it's actually super simple. It's just the number of openings or meshes you can count in a 1-inch line. So a number 10 sieve? Yep, you guessed it, 10 openings per inch. It's a really simple, elegant system.
But it's not just about how many holes there are. Not at all. These things are precision instruments. To make sure every batch of medicine is consistent, engineers define literally everything: the exact size of each opening, the thickness of the wire itself, the total open area for sifting, and even the tiny, tiny acceptable margin of error in the whole size. Every single detail is standardized. It has to be.
The material the sieve is made from is also a huge deal. I mean, sure, you could use iron, it's cheap, but it rusts, and that rust can contaminate the medicine, which is a massive no-go. And that is why stainless steel is the gold standard. It costs way more, sure, but its amazing resistance to corrosion makes it the absolute best and safest choice for pharma.
So what about other metals? Well, pure copper is pretty good because you avoid any iron contamination. But it has its own problem. It's a soft metal, so it bends and gets distorted really easily, which makes the sieve totally inaccurate. The solution? Use copper alloys, like brass and bronze. They have that great corrosion resistance, but with way more strength, so they actually hold their shape.
And you know what? Sometimes the best solution is to just get rid of metal completely. Synthetic fibers, like nylon, totally eliminate any risk of metallic contamination. But here's the really surprising part. You'd think plastic would be weaker than metal, right? But for creating these superfine meshes, these synthetic fibers can actually be stronger than a metal wire of the same thickness. How cool is that?
Okay, so we have our precision-engineered sieves, made from the perfect material. Now, how do we actually use them? It's time to check out the different methods for putting these tools to work. In a factory or a lab, you're not just going to see someone shaking a sieve by hand. There are three main mechanical methods. You've got agitation, which is basically just shaking or vibrating the powder through. For powders that are a bit sticky, you might use brushing to keep everything moving and stop the holes from clogging. Or, for a really high-speed approach, there's the centrifugal method, which spins the powder and throws it against the screen with huge force.
But here is the single most important rule, and this is a direct quote from our source material. Material should never be forced through a sieve. Sieving is all about separation, not brute force. The whole point is to let the correctly sized particles fall through on their own. If you try to force them, you could damage the sieve and completely ruin the entire process.
But what if you need to separate powders that are unbelievably fine? Or what if you want to avoid using a physical screen altogether? Well, that's where we get into some really advanced methods that go way beyond the mesh.
This is where a process called sedimentation comes in, and the principle behind it is actually really simple. In a fluid, like air or water, bigger, heavier particles fall faster than smaller, lighter ones. It's just physics. And by controlling that process, you can separate particles by size without them ever having to touch a screen.
And this right here is one of the coolest applications of that idea, the cyclone separator. I know it looks a little complex, but the idea is just brilliant. You shoot a mix of air and powder into this cylinder at high speed, which creates a crazy vortex inside. That spinning motion throws the heavier solid particles against the walls, and they slide down to the bottom to be collected. All the while, the clean air just escapes out the top. It's such an elegant way to separate a solid from a gas.
So when you step back and look at it, we've gone from the complete chaos of a raw, mixed-up powder all the way to the incredible precision and control you need for safe medicine. Size separation really is a critical journey. It's about taking that chaos and creating perfect order to make sure what's supposed to be in your medicine is actually in there.
And that kind of brings us to our final thought. The next time you open a bottle of pills, or you see a simple scoop of protein powder, are you going to think about all the complex engineering and the careful science that was required just to make sure that simple powder is safe and that it actually does its job?